Welcome everyone. In this video, we will learn how to design primers for polymerase chain reaction. As we all are aware, PCR reaction helps in the amplification of DNA or making more number of copies of DNA. And for setting up the PCR reaction, primers are one of the important components apart from tag DNA polymerase, gene, DNTPs, buffers, and any other cofactors. Now, for designing the primers, both the forward and the reverse, I need to know the sequence of the gene. And for this particular video, we have selected the spike gene of the novel coronavirus. Now, for getting the sequence of this spike gene or gene S, we'll go to the NCBI site, that is the National Center for Biotechnology Information. So we'll go to the NCBI site. And what we'll do, we'll go to this tab where they have a separate section for SARS-CoV-2 data. We'll click it here. And then we'll scroll down. We'll see, we can see the view record, the NCBI RefSeq SARS-CoV-2, that stands for Severe Acute Respiratory Syndrome Coronavirus 2 Genome Sequence Record. And as you can see here, it shows Severe Acute Respiratory Syndrome Coronavirus 2 Isolate genomic sequence from Wuhan. You can read all the important information here. It shows like it has 29,903 base pair single-stranded RNA, the family name, organism, source, authors who have published the first, like the sequence of this genome first. You can go down and can have a look. But remember this particular FASTA sequence, or if you are going to have a look at these sequences, this is going to be the entire genome of novel coronavirus. But what I'm interested in is knowing the spike gene sequence. So I will try to find it out by looking for the S gene. So let's try to find it out. And as we can see here, it shows gene that corresponds to the gene S, because that is our major objective. I want to design the primers against this spike gene, which codes for the spike glycoprotein as a structural component of the novel coronavirus. I will just select this brown highlighted region by clicking on the FASTA sequence. So here is the sequence, fine. Now, how we are going to design the primers? The one of the tool which we are going to use is the NCBI Primer BLAST itself. And you can reach there by clicking on Pick Primers. Fine. So it has automatically taken the accession number. If you're not sure about the accession number, if you're not getting those accession number from the NCBI, you can still copy those FASTA sequences and can paste it here. Now there are a couple of information which you need to read it. It says the forward primer should start from 21563. Fine, because that is the reason from where the spike gene starts. The reverse primer, like the forward primer and the reverse primer, should be located from this to this. Fine. If you do want to amplify a specific region within these fragments, you can give those numbers. So like you can see the sequence of the spike gene, and if you are very much particular about a particular sequence, you can give those numbers. You can select those numbers and you can mention those numbers. For this video, we'll just keep it as it is. I just want to amplify the region which is lying between 21563 and 25384. Fine. Now, do you want to use your own forward primer? No, I don't have any such sequence. So if you have designed manually, you can keep it there just to look for the properties and the melting temperature or the annealing temperature. It's telling like this section is for the PCR product size, minimum 70, maximum 1000. So that's fine. I mean, you can just a bit increase the minimum product size. So this is the PCR product size which you are going to obtain. You can keep it 70, that is the by default, or you can change it to something which is higher than the 70. So I have kept it at 2000. You cannot not go beyond 1000. So that's fine. Primer melting temperature, minimum 57, fine. Optimum 60, maximum 63, seems all right. You can gather the information by clicking here. So the TM calculation is controlled by table of thermodynamic parameters and salt concentration formula, and the default table of thermodynamic parameters and the default salt concentration formula. Here is the link as the recommended by primer 3 program. So you can read these informations by clicking on this question mark. I just want to keep the difference. I will just lower it. So I will keep it 2. So what I'm trying to say, that the maximum TM difference should be not more than the 2 degrees Celsius. Exon junction span, these are like the simple sequences. Remember, they have a 30 kilo base pair plus strand RNA sequence. That means it is going to get directly coded to form the protein. So I will just say not much preference, no introns inclusion. This tab is for those sequences where do you think that the exons are interrupted by the introns. So you need to make sure that I'm designing the primers depending upon your interest, whether you want to include only exon junction, or it may not span exon junction. So this holds true for if the sequence is carrying the introns. As I said, the entire sequence of the novel coronavirus is quite simple. It gets directly coded into the protein. So that's fine. A specificity check. So read it. Search mode automatic. Fine. I don't want to guide it. Database. So I will select here the RefSec representative genomes. What does it tell you? It tells you that this RefSec is a database of annotated genomic transcript as well as the protein sequence records. If you want to study, you can click it here and can read. So the RefSec representative genomes contains NCBI RefSec reference and representative genomes across broad taxonomy. That's fine. That's what I want. Like they can look for the similar sequences in the family. Organism, remember these coronaviruses or novel coronaviruses comes under the family Coronaviridae. I hope you all know that Coronaviridae, and we'll just select this taxonomy ID. One should know, like if you are dealing with the proteins from the humans, then you need to select Homo sapiens. If you are dealing with the proteins from the rat, then you need to select the organism as a rat. I'm just selecting it as a Coronaviridae because this novel coronavirus comes under this family. That's all right. Primers must have at least minimum, fine, 2 mismatches, fine, at least 2 matches. I mean, fine. So this is all talking about the stringency. Again, you can click it here, read it here. This requires at least 1 primer to have a specified number of mismatches to unintended targets. The larger the mismatches, as we all know, especially those towards 3' end, which is not a good idea, are between primers and the unintended targets. The more specific the primer pair is to your template. Fine. That is, it will be more difficult to anneal to unintended targets. However, specifying a larger mismatch, you may make it more difficult to find such primers. Try to lower the mismatch value in such case. That's what. You can even reduce it to 1. We'll keep the values as it is. Maximum target amplicon size 4000. This specifies the maximum amplicon size for a PCR target to be detected by primer blast. In general, the non-specific targets become less of a concern if their sizes are very less, since PCR is much less efficient for larger amplicons. That's fine. I will leave as it is. So results in a new window. That's fine. I am just looking for the other parameters. The PCR product size, like we have changed it. Number of primers to return. So that is telling me that they are going to give me the hits of around 10 primers. So Seems all right. I'll just click it on the get primers. It's going to take few seconds. It's going to take few seconds. So suggesting that it's submitted, it's going to take few seconds. I've already done it for you. So I'll just come back to the results parts. What you will get after few minutes is a window like this, and have a look. They have given, you know, the different primer pairs, and as you can see it from this picture, you have these many pairs: primer 8, primer 6, primer 10. That's what we have selected, 10 primer pairs, and the information is given here. So you can go and have a look at these sequences. Now we'll just focus on the first one, where they have given the sequences for the forward primer, reverse primer. The product length is somewhat close to 613, and the length of the primers, starting, stop, melting temperature seems fine. There is not much difference. They are very close, which is a good thing. GC percent seems okay. It's a 60-55 for the reverse primer. The self 3' complementarity 3 and 2. We'll learn more about these in a moment. So now these values are there. These sequences are there. I need to check the other parameters like hairpin, primer dimer, GC clamp, and other things. Like one of the very important thing, that it's always good to include G at a 3' end, because that either G or C at a 3' end. Why so? Because it gives a stronger binding at a 3' end. Remember, the G pairs with the C with the help of 3 hydrogen bonds. So it's always good to have these G and C at a 3' end, because that will result in the stronger binding, and thus the extension by the DNA polymerase from 3' end. Okay, now we'll just have a look at the primer stats values. So what I will do, I will click on primer stats, where we will learn more parameters. PCR primer stats, as you can see, you can click it. And here I will type my forward and reverse primer. So, sorry, I will type the forward primer. We just need to copy it. I have already taken the sequence on this Word file, so I'll copy it from here. Come on. Remember, this is the same sequence GACTA, which I have obtained it from here, GACTA. Reverse primer. Remember, these sequences are in the order of 5' to 3'. So Command C. Initially, I copied it in the Word file, so that's the reason I was copying it from Word file. So this is about the forward. Now go back to reverse. It's always good to type this forward and reverse, because this is the way they read it. I will click it here, 5', 3', 5', 3', and then submit. Now have a look. This is very important window where you'll learn many things. General properties, they have given primer name, primer sequence, fine. Sequence length 20, it's okay. You can even try to increase it by including these restriction sites at a 5' end. Remember, it's always good to have the restriction site at a 5' end, because that will facilitate the process of directional cloning if required. Base counts fine. GC content as discussed 60 seems okay. Molecular weight, nanomole amount given 5.44, good. What we are interested in is the nearest neighbor TM, which is close to 64. Remember, this is the melting temperature. So annealing temperature is going to be -5 of whatever the TM value is. So that means close to 59, 60. Good. Single base runs. So there are no more repeats of any nitrogenous base. That's the reason. That's the thing you can see here is showing pass dinucleotide base runs. That means the repeated nucleotide, which is not a very good thing, because that can affect the ATGC content as well as the slippage. Length pass, percent GC pass. TM is greater than 58. I mean, no problem, 58, fine. Which they are talking about this value. GC clamp, warning, there are more than 3 G or C. So as you can see, there are many more G. As I said, it's good to include G or C at a 3' end, but we should avoid, the nucleotides which are in a long stretch, because as I said, it can create a slippage or it can affect the GC sequence. So what you can do, you can just try to emit 1 or 2 of these GC, but I don't think so that is going to affect much more. So self-annealing pass, hairpin formation pass, that means it's not creating. There are many other analysis tools. One can even use oligo IDT. I'm using here the PCR primer stats. How about the general properties of the reverse primer? Seems okay. Sequence okay. Sequence length fine. GC content all right. Molecular weight nanomole microgram. These are the values which will be required for setting up the PCR reactions. Again, 64.69 nearest neighbor TM, fine. 64.9 seems all right. So you can see, the properties, all of these parameters are showing pass, pass, pass. That means good. The TM is greater than 58. That's all okay. They are giving the warning, but this is not very serious, because if the TM is more than the 58, which in this case is the 64, what is the annealing temperature going to be? Close to, so it's close to 65. So the annealing temperature is going to be close to 60. Hairpin formation pass. There is one warning where they are showing self-annealing, which should not be a problematic situation. Why? Because have a look at their orientation. It's a 5' end, it's a 3' end, it's a 5' end, it's a 3' end. So, there is no possibility of extension from this side. Same here. It's the 3' end which gets extended from this thermostable DNA polymerase. So no need to worry about. And also, these are going to form at a lower temperature. As soon as I'll increase the temperature close to 60, these will be broken down. And nowadays, we make use of hot start protocol, touchdown PCR. So those things will further eliminate these problems. So seems all right. We have designed our PCR primers, and if you can see, which I have written here, like these are the same 20 nucleotides, forward, reverse. Remember, some of the times, as I was discussing, many of the times you need to incorporate restriction enzyme sites, and we do incorporate at a 5' end. Let's say if I want to incorporate EcoR1 site, GAA ETC. So fine, I can incorporate it, and these are not going to affect the melting temperature much, mind it. These are not going to incorporate melting temperature first, but what I'm trying to say is that one can also include, the site for a different restriction enzyme at the reverse primer. The only thing which you need to know, after adding some of the sites for these restriction enzymes, once you have added, make sure to add few extra nucleotides at a 5' end. Why? Because these restriction enzymes are protein. They need to cut these sites, and they do these digestion. They do recognize this sequence and cut it by sitting it, by requiring some space, and that binding space I'm providing by adding few more nucleotides. So let's say ACG. So let's simplify it. This was the sequence. This was the sequence, the one which is highlighted in black. Then I have added a site for the restriction enzyme that you can choose whether you want to incorporate EcoR1, BAMH1. There are multiple sites, but be very careful. The sites which you are adding should not be there inside your gene of interest. Fine. And then I'm adding some 3-4 more extra nucleotides, because that will be acting as a sitting place where the restriction enzymes can come, bind, and then can act on these sites. Remember, these are proteins. There needs to be a binding cavity where they can sit, and they can act on these sequences. Same for the reverse primer. If you are using some of the sites for the restriction enzymes, just don't forget to add some 3 or 4 nucleotides at the initial 5' end. Remember, these are the sequences which are going to bind to the gene of your interest and not these. And these extra sequences coming from these restriction sites, these are not going to affect much of your melting temperature. And also remember, since in the initial few cycles of these sequences are not going to bind to your gene of interest, I mean, you should not be bothering too much about the GC content and the annealing temperature, which should be coming from this region. I am saying that was the reason, like, we calculated those stats only from these sequences. If you want, you can calculate from the entire sequences, but that is not going to affect the scenario much. I hope this part is clear to each and every one of you. If you do have question, feel free to write it in the comment box. And thanks everyone once again.